All right, everybody. Sorry about wrenching you out of your breakout rooms, but hopefully everybody had a chance to get things wrapped up. Um, we will go ahead and get started in just a minute with the final presentations. Um, just wanted to thank everybody for participating in this year's hackathon. We're excited to see where your simulations ended up and what you plan to do with them after the hackathon. Um, Just as a reminder, this session is being recorded and live streamed to YouTube. Um, and we'd like to start by thanking our funding sources through the NSF, as well as our support networks in the IUB Biocomplexity Institute, as well as the OpenVT and Glimprint initiatives. So I think if people are ready, we'll just go from the top of the list.
All right, so we are team heterogeneity. And so as you can see in the background, I've used some pixel art because CompuCell3D is basically, there's a lot of pixels and stuff going on. So I thought that would be interesting. The font is kind of pixelated as well. So this is the team, Shivani Agarwal, Shamaita Roy, Jamna, and then there's me. And so the introduction, as you can see, tumor invasion is governed by the balance between cell-to-cell adhesion and cell ECM mechanical interaction. Cell ECM coupling provides, yeah. So basically, tumor invasion, the one that we are investigating here is, uh, so between cell to cell, there's some adhesion, and there's some adhesion between the cells and the ECM. And we're trying to investigate, well, how does the heterogeneity in that adhesion between cells and various ECM cells lead to the migration of the tumor cells, whether they be single or collective? All right. So the hypothesis here is that, we have tried introducing dynamic adhesion using the FPP link plugin, and so using spatially heterogeneous FPP mediated cell ECM interactions, we basically try to see how the cells invade compared to the adhesion flex approach that we took last time. So, and how that leads to altered invasion. Uh,
Yes, so basically what we are trying to do is we're trying to introduce dynamic and spatial heterogeneity in FPP links. So the cells at the boundary are more prone to have a link with ECM as compared to cell at the core. And the links are keep on altering as the ECM is degrading and remodeling, that links keep on deleting or adding. Therefore, we can see the extent of invasion by the interaction between ECM remodeling and FPP links, and how these two get affected in a situation where there is only cell cell adhesion and there is this dynamics of cell cell adhesion, ECM remodeling, and FPP. Okay, so in this, we are just generally explaining how a tumor starts to invade, starting from its primary sites. It's degrade the ECMs, try to go towards the blood vessels, and from the blood vessels goes to the extravasation and try to find a secondary site where it can colonize again. So our primary work based only on this dynamics, where we are seeing how the interaction between ECM fibers and the boundary cells through the links can affect the invasion. For that, we have this paper where they have experimentally proved that the focal interaction or adhesion due to the fibers between cell and ECM can increase the extent of migration and invasion. So we have represented that with FPP, where FPP can be interpreted as local mechanical coupling anchor between cell and ECM. and how that anchor helps the cell to physically pull towards the ECM and alters the migration and invasion.
As previously mentioned, we have used the focal point plasticity plugin, so which is basically a core screen representation of the mechanical coupling between a cancer cell and its neighboring ECM. In a real migrating cell, movements involve a coordinate sequence of events, that is, acting polymerization is producing protrusion, new adhesions are forming, and myosin generates the contraction, and the forces are transmitted through these adhesion machineries to the ECM. This produces traction and the ECM deformation, allowing the cells to move towards, followed by a disassembly of adhesions at the rear. Now, FPP represents this whole mechanical sequence as a simplified mechanical link between the cancer cells and the neighboring ECM cells. So, in our model, the link is very dynamic. It can form where cancer cells encounters this ECM, and it can be lost when the cell is moves away, or when MMP mediated ECM degradation degrades the ECM, and thus the link gets deleted. Therefore, FPP, it allows us to capture this dynamic cell ECM mechanical interaction, while MMP controls this remodeling and the availability of the ECM itself. Now, this combination allows different cancer cells to experience different local mechanical interaction with the surrounding matrix. So now the framework of this whole project is that the cancer cell is interacting with the surrounding ECM through these links. Now they are providing the mechanical coupling between the cell and the ECM. At the same time, the cell is also producing MMP, which is degrading the ECM. So now, as MMP is remodeling the ECM, the collagen that is available for cell ECM interaction, it changes. So because of that, the FPP attachment of the cancer cell is lost, and a new interaction can be formed with another available ECM region. So the FPP interaction is also incorporating the cell's mortality. In our implementation, the cancer cell movement contains a random component as well as an FPP dependent component. So the local cell ECM interaction, it contributes to the direction of the movement. Therefore, this model basically processes cancer cell motility, then MMP mediated ECM remodeling, and this dynamic cell ECM mechanical coupling through FPP.
Here we have drawn the analogs between the experimental findings and our interpretation. And our final result is this, where the figure A shows the invasion extent when there is no FPP. Only the cell-cell adhesion is controlling the extent of invasion. And we have the second A is basically how we have done the image analysis, where we have converted this into a black and white image, and then we calculate the area parameters, circularity, solidity, all these parameters to check the extent of invasion. The B is the one where we actually have the FPP links, and we can say that even in the same parameter set, but because of the FPP, the tumor are more invasive as compared to before. And then we have drawn these parameters, which is area parameters, circularity, solidity, number of clusters, maximum invasion distance, and mean invasion distance. And all of these parameters thus depict that because of the presence of FPP, we do see an increase in invasion as compared to a system without FPP. And in the future direction, we will try to incorporate two models that I'll show you in just a minute. The previous one that we are trying to do is an integrin based uh, adhesion flex coupling between cell and ECM, which we failed to actually conclude. And that's how we start to do this current model if we are with the FPP links. But if it's possible for us to link this integrin based adhesion flex and FPP links together to see how the tumor microenvironment can uh, help the cell to invade as compared to studying particular cell. So the simulations. So here as we see that as the simulation goes on, the color changes. The color changes depict that the connection keeps on adding and deleting as the ECM is remodeling and as the ECM is degrading, the cells try to find the nearest neighbor ECM to connect with, and hence there is a change in this color. This is a simulation with FPP links. We can see in black the links between cells and ECM. And let Now I'll show you the simulation without FPP links. Here the extent of migration is less. Cells are more adhered to each other than to ECM. And in last 30 seconds, I also want to present what we are earlier trying to do, which is the intriguing modeling, where we have a different intriguin concentration on each cells. The intriguin are the molecule density that bound the cell to ECM, and the cells at the boundary have more integral concentration. That's why they are more red and orange in color as compared to cell at the core, which is why they are more in blue color. But we don't observe a maximum ECM. Okay, but we don't observe much invasion in this because instead of migrating through the ECM, the cells are stick to the ECM, hence there is a decrease in migration. But if we couple this with FPP and we introduce traction force and direction, maybe we can observe more invasion by combining these two models together. Thank you.
All right, thank you. Feedback from instructional team.
I'm putting my observations on the chat. My main question is whether the cells in the center are also creating links with the ECM.
Yes, we do observe that, but then we try to like study particular cell, and we do see few cells that are completely surrounded by other cells do not particularly have those links. Maybe if I run this again.
Remind me again what the color changing represents.
The color change basically represent attach and detach of ECM. Okay, it's deleting the previous connection and then again trying to connect with the nearest neighbor.
Okay.
So right there, we see this link connection in more or less the center of the lattice, a little bit up, a little bit to the left. It is connecting a piece of the ECM on the left of the cluster to a cell that is in the center, more or less in the center of the cluster. What is this link representing? Does the cell has a philopodia extending all the way from the center to the ECM outside?
No, not philopodia.
Is this perhaps an artifact of the method you were using?
Maybe yes, because we did try to print every command and try to see if there is connection or not, but we don't see an actual connection in that. And here, maybe because the cells are migrating and the blue here did show ECM because this is not cell.
Mhm.
Maybe because
Maybe,
of that.
maybe you could rerun this, just showing the cell field, not the FPP visualization field.
Yeah, reruns. So the green is basal membrane, blue is ECM. This is the MMP secretion, and as they secrete, it's tied to degrade ECM.
I'm sorry, when you say this, I'm not sure what you're pointing at. What is the white? Is that degraded ECM?
So what we are doing is we are as the MMP secretion reaches to a certain extent greater than threshold, cancer ECM represented at cell here, C1 converted to C lysed, then there is a decrease in volume of C lysed, and as it decreases to a minimum volume, it converted to a NC1. NC1 is just a blank space available for cancer cell to occupy so that they can migrate through the degraded ECM.
Okay, so you have a bunch of different materials that are represented as the extracellular. Fine, that's great. It's a very interesting model. I'm not sure why you produce so many links to particular small patches of the ECM when you run.
Yeah.
I would have thought that the cells would link out. When you do your link calculations, do you have the source of the links be the cells or the ECM? If you
Source is
run
cell.
this, okay, that I don't think that can be right.
Oh,
I don't think that that's just, if you just why don't you keep running it for a second and hit pause.
okay. So should I increase
No, just keep
MCS?
running it. Just keep running it. Okay, or just run it again. Just run it again.
Yeah.
Don't change anything.
Oh, I'm just increasing number of steps.
Okay. Could you pause it, please? Okay, so if I look at the zoom in a little bit on the bottom center. Okay, so you have many, many cells all binding to the same extracellular matrix region. Why is that? I'm not saying it's wrong. I just want to understand why
Because
you're doing
they are
that.
trying to find the nearest neighbor ECM.
So that they bind to whatever ECM is closest to them,
Yes.
whatever that is. And all of the cells do that. So even the cells at the very center are binding to the ECM, the very long filopodial attachments.
Maybe they are not getting deleted because what's happening initially, if we have 16 cells and as they proliferate, the cell initially present at the boundary remains, stays there, and as they proliferate, it won't be able to come out of that initial position. So the links
No, that's
remains, but
definitely
it's
not
not
what
getting.
that's definitely not what's happening because you're deleting the links and reforming.
Yeah.
So your algorithm for forming your links maybe is not doing quite what you think. You have a lot of really good things here, so I'm very impressed with what you've got, but I think you may have to think a little more about the algorithm that you're using to generate your links and make sure this may be what you want, but if you don't want cells in the center of the aggregate binding to the ECM, then you're going to have to change your algorithm, maybe have a maximum link length, for example, or only have cells that are in certain regions form links. So,
Okay. Like for example, this cell is not having any link. So
yes,
I'll try to improve
but
my
the one
code,
at the very center is,
but this one is having.
yes. And they're also all binding to the same ECM volume.
That's because the nearest neighbor. I have like introduced, try to find nearest neighbor.
So I think that nearest neighbor, maybe you need to relax a little bit about that.
Okay.
But it's a great start. I think it's very impressive that you were able to do so much in 3 days.
Thank you.
Hey, tell us what you've been up to. Look forward to hearing about it.
Okay. In my project, I attempt to model a growing tumor that at a certain point connects to a blood vessel to receive nutrients from it. Biologically, my entire simulation is flawed, as I set parameters such as field diffusion and growth rates to exaggerate levels. In my simulation contains 4 cell types. P, the blue one here, representing the tumor cell, N cell, representing the necrotic cell, and V, the yellow one here, representing the neovascular cell, and the EC, the red one, representing the endothelial cell. Regarding for the chemical fields, I implemented 3, the glucose secreted by EC cells here, and the fooling tumor growth, LVEGF secreted by the tumor and promoting EC and NV cell growth here. And the SVEGF secreted by P cells and acting as a chemical signal for EC and NV cells here. I realize that there is much to change and would appreciate feedbacks for improvements. However, this project primarily served as a way to learn how to use CompuCell and attempt a slightly larger, more complex project. Furthermore, I hope to use this knowledge and any feedbacks provided to create a better angiogenesis simulation in the future. The necrotic cell I included was intended to represent the result of glucose deprivation in P cells. However, because I set the glucose field level so high, this outcome does not actually occur in the cells. And that's it. Finally, I would like to thank the instructors for their patience and commend their workshop. It was certainly a great experience, and I learned a lot over the past 2 weeks.
That's great. I'm glad that you got this to work the way you wanted to. I see that the endothelial cells are proliferating in a way that endothelial cells probably wouldn't proliferate in real
Yes.
blood vessels.
Yes.
So you might want to think about regulating the proliferation a little more if you were going to refine the model that you started with.
Yes, of course.
But you have the base tumor model with the signaling and the nutrient transport working very well. You have induced proliferation working. You have the vascular structure working. So I think you have a good framework for developing additional methods.
Thank you.
For your main project, do you want to do cancer simulations, or are you interested in other things and the cancer was just an interesting way to learn?
I want to model a tumor angiogenesis.
So you do want to continue with this direction?
Yes.
Well, I definitely would like to see that cancer model made more realistic.
Yes, I will provide the results and show to you.
And anything we can do to be helpful, please let us know what we can do.
Okay, thank you. I appreciate it.
Thank you. Thank you for sticking out the course. I appreciate it.
Bye-bye.
Yeah, thank you. Bye.
Thank-
Boundary conditions apply. Clara Lauder.
Yes. Hello, can you hear me? Hi.
Yep.
Cool. I'll screen share. So yeah, thanks everyone for helping and being here. It was really fun. I mean, I worked with Pedro quite a lot, but it was still fun to see everyone again and get some simulations very quickly into shape. Yeah, so this project was a sub-project of what we are working on with wound closure, which is the two GIFs you can see, and that really investigates how the boundary conditions and domain size affect the outcome of a simulation of this type. So in the wound healing, if it closes or not, and how fast it closes. And we investigate two different boundary conditions. One that is, we call it compliant. So that's the right-hand side GIF, where it moves in with the cells, and so it will always close. And the other one is a solid boundary condition, or we call it non-compliant. And there, it won't close if the domain is too small compared to the wound. Mainly because the cells need to stretch to close the wound, and they can only do this so much. So that's what we investigate in our main project. And the idea was for the hackathon to make two sub simulations, two other simulations that demonstrate the same effect. And one of that is the tissue folding that I worked on, and the other one is an immune simulation. So in the tissue folding, how do I, here. In the tissue folding, the concept is quite straightforward and the same as in the wound healing, because you have a boundary on the left and the right-hand side of the tissue folding simulation, and there's only really a string of cells. And when they fold, the blue cells in the simulation, they have to move in with the fold. So they can only stretch so much. And so that will hinder the folding if the string is attached to the boundary. And that's similar to the non-compliant wound healing simulation. So if the domain is too small, it won't fold, and that's what you see in the top. So initial condition, and it starts folding. You read this from like top to bottom, and it can look like this, or sometimes it can also look quite crazy, but it won't fold if the domain is too small, right? But if you make the boundary condition compliant, so in the case of tissue folding, it's just you detach it from the boundary, then it will always fold. So that's a demonstration of that effect. And then if it doesn't fold in a small domain, we can make it fold in a larger domain, but just increasing the domain size, and we can demonstrate that as well. And the screenshots do demonstrate that. And it folds nicely when you set the domain size large enough. All right, so that's the comparison to wound healing with the tissue folding. And we also did a little detour where we investigated something about the tissue itself, which was we made a string of tissue, and we just applied a force on the right-hand side that was pulling the tissue to the right, and it was attached on the left-hand side. And we investigated basically how the force dissipates into the tissue, or more like the length of the links between each cell. And we vary the contact energies between the cells, because we actually had trouble getting it started to fold. And what we saw was actually that, this is more of a side note, but you can really see quite clearly with these chimegraphs that the light green region, so the larger links, don't dissipate into the tissue so much when you have low contact energy, so when they're really tightly bound. But when their contact energy is quite high, then yeah, it dissipates better, the link lengths. Right, but we actually can't go with the right-hand side, so we fixed it, but there's a reason that we didn't like this case, and that was that there's actually the cells themselves have a spring energy to them. So they don't get the stimulation doesn't start in neutral energy, in relaxed state, because they have, so they're of how we see the cells in their size, like the cell target volume, and then how they are kind of pushing away from each other. Right, so we want the system to be relaxed for these kind of measurements and these kind of observations. Okay, and then the second thing I was building in this hackathon was an immune simulation. So there is a lattice of healthy cells, the light blue in the simulation, and they get infected by one initial cell that's infected, the red cell placed in the center. And then the infection spreads probabilistically, so some probability to the neighboring cells. And we also place some number of immune cells in the domain, and those are the tiny purple cells. And they can move, and they will move towards the gradient that is highest of the immune cell secreting some sort of chemoattractant. But they move randomly, so this is random motion with a bias towards the chemotactic gradient. And I mean, there are a lot of other rules here, but the basic idea is again that we want to see how the domain size affects the outcome here, and we want to find a domain size that is large enough so that the outcome is not affected. So if we would measure the size of the infection, then we need to choose a domain size where the number of immune cells, which scale, okay, so the number of immune cells are seeded per lattice pixel or per domain size area. And so if we increase the area, then the number of immune cells scales with that. And so if you have a really small domain, and I'll show this here, if you have a really small domain, this is 50 by 50, the top picture, then you have fewer immune cells. And if you increase the domain size, then it scales by that increase. And so you can see the difference in how the infection is fought. In the top picture, it can spread, in the bottom, it can't. And I think I can't see the timer. I don't know how I'm doing for time, but the idea is the same again as in the wound healing case, where if you choose a domain size that's too small, then your infection spreads and covers and infects the whole domain. But if you choose a larger domain size, yeah, it won't spread. And if you want to measure the area of the infection, you don't want to have your results skewed by the insufficient domain size or wrong domain size. But this is really more an illustration of the fact that this effect exists. And for the wound healing simulation, we actually have concrete measurements of how you can measure which domain size you want to be in. But for these two toy models, we haven't gotten there. So yeah, I think that's all.
What are your plans going forward?
Yes, so I think we have to fix a few technical things with the simulations and discuss a few more things like that. And then we'll have to see how exactly we're going to tie this into the existing work for the wound healing for publication. But these are really just two demonstrations of this effect, so it's not too big of an effort. Yes, and going forward, I mean, I chose this immune simulation to build this because we actually want to overlay this on the wound healing. So going forward for a different project will be to merge the wound healing with the immune system and add a lot more detail and a lot more realism, and then study some immune effects on the wound healing. But the immediate goal is definitely to just put that as demonstrations into the publication.
Again, I said it before, I'll say it here. in front of everybody. I think it's really exciting to have the coupling between the CompuCell and this cellular automaton model. There are a couple of groups this year that have tried to couple between different kinds of models. And to me, whenever I see somebody do that successfully, I find it really exciting. I hope we could somehow get that into our code base, or at least teach people how to do it, so people know how to do that cross-method coupling.
Yeah, it's been quite effective. We chose the cellular automaton on top because the immune cells are much faster than cells in CompuCell. So we wanted them to be able to move really a pixel at a time for each Monte Carlo step, which was necessary here. So yeah, I think I forgot to mention that they are actually cellular automaton cells. Great.
One of the possible applications of this, James, is so usually you will use a statistical method for finding a large enough domain size. So you have your metric, you increase enough, and then you do a sensitivity test. But that's very expensive. So the other way is you identify the scales of your system, the time and size scales of the system, and then you mechanistically infer what is the minimum domain size for that. So for example, in the wound healing, from the kymograph, we can calculate how much time the signal travels to the border and back by fitting a mechanical model there. And then we use this time to define, okay, if we set to this domain size, we are safe. So we are thinking about using the same strategy for these two other models. And then if we show that this mechanistic inference of the minimum domain size works for these two models, we show that there is this alternative mechanistic way of finding cheap way of finding the minimum domain size that would bypass having to run these heavy statistics situation.
That's a great point, Pedro. I think it's exciting work, and I'm looking forward to seeing how it goes forward. And having the fast immune cells would have helped us a lot. You could have explicit bacteria, you could have explicit virions, as well as immune cells. Great method. Look forward to seeing you two work together. Okay, who's next?
The Hackathon group.
Okay, I think I have it up. Y'all can see the presentation, right? Cool.
Yep, looks good.
So I'm Kyle Hawkins. Joining me in presentation is Nate Lesage, and Yannick couldn't make it today. He had some things he needed to do. He may or may not be listening in, but yeah, so I'll take it from the top. Our presentation is about us working on a cellular POTS model for dental plaque and trying to understand some of that spatial structure and the pH dynamics. This is our project for the CC3D workshop Plaqueathon of 2026. So within our project, we were interested in a couple questions. I was mainly interested in how growth was affected by having spatial differences of pH, and I was curious about whether growth, or if you put that feedback into growth, how that would affect pH microenvironments. Yannick was interested in the effects of how copper would affect the system, and Nate had an interest in the demineralization process. So those are the different interests we had for this project, and kind of showing a overview of the modeling process and how these all connect. We basically all have this core of chemical fields interacting with biomass being the different bacterial guilds, and then pH. So metabolism connects the chemical fields and the biomass. Basically growth terms due to the pH affect the biomass term, and then the different pH chemical compounds or different acidic compounds pronation states are affected by pH as well. So for our different hypotheses that we're interested in, I was interested in the arrangement part. This is where copper comes in, basically affecting the fields, and also the biomass. And then for demineralization, the pH causes the demineralization of enamel, and that's where that comes in. So we were all working towards developing this core here. As far as the bacteria goes, these are the ones that we chose to model. So this is from a paper by Ely et al. And they did a one-dimensional model of how these different bacterial guilds affect pH dynamics with a glucose impulse. So this is the information for it. This is the mass percentages they had, and for the model, these are some parameters I used to represent basically favorable pH ranges for each of these different groups. For our problem, this is the geometry we used. We had a saliva level up here, a tooth level down here, and then a plaque level where Yannick was able to get a nice random initialization of all the different bacterial species inside of our lattice. So this is our geometry. As far as chemicals fields go, this is the full list of the different chemical species from the Eiley paper. We didn't use all of these. The ones that we did use are the ones in black. We've taken the words of advice to start simple. This is what we chose to start with, rather than dealing with everything. The thing, and including more of these things is a clear extension of this model, but we chose to start with this group of chemical compounds. A good majority of them are acidic. They have different pronation states. The different pronation states also have different diffusion constants. So in order to model the diffusion, we had to have every single, I guess, chemical compound present in the model for the fields. And that's the chemical transport part. As far as metabolism goes, we chose to remove aerobic processes and the processes that involved polyglucose. So that left us with three different processes. One being a metabolism of glucose at when there's a lot of glucose present, that being glucose turning into your lactic acid. And then for low glucose, we had a different reaction process. And for one of the species, I think VEL, they had lactate fermentation. So that gave some, I guess, connection between the different species, as this species would turn this more acidic acid into less acidic acids. And this is a visualization of the network. Metabolism, this is the rate laws we used. We have different nod terms, inhibition terms, and we have a pH term for this. So this is the equation we use for metabolism. pH, this is how we calculate it. Basically, we did a charge balance, and then saying that residual equal to 0, we were able to find the pH for each last site. And then I guess Nate wanted to talk about the growth part of this model.
Yeah, so I wanted to implement cell growth by using this model that we found in the literature. It's called Michaelis-Menten kinetics, and I saw this in several papers where they were using this to model the growth of the of bacteria. And so I found the parameters that we would need for each species of bacteria, and was in the process of implementing this. And I got almost all the way, although just due to lack of time constraints, there was one parameter I wasn't able to determine. So we're still working on the cell growth as of right now.
Yeah, and I guess here's the Docker.
And I also worked on building a Docker image for running CC3D, since I was not able to build the CC3D project on my laptop. So what I was able to do was create a Docker file, which and build run a Docker image off of that file, which has a lot of advantages. So one of those would be reproducibility, because you are able to reproduce and you're able to run the Docker image in any environment that supports Docker. So if you, because what Docker is it's kind of like a virtual machine. So you're able to reproduce the same image on different environments. And also it's nice because it's a container, so everything that you put in the Docker file will be runnable on the image. So if you have, if you know the steps to basically install and create and your environment, then you can write that in the file.
So
will be there every time. And it's also running on any desktop or any system that supports Docker. So what we have is we're able to run the Python API and Jupyter notebooks, which you can access in the browser through the Docker image, which is really nice. And we're working on the actual CC3D GUI itself, because Docker does not have out-of-the-box support for running GUI applications like CC3D. Takes a little bit more setup.
Yeah, it's really cool that you were able to set this up, Nate. And as far as simulation stuff goes, I got a version of the simulation without the growth part of it, which obvious next step is to get growth in there. And so I'm able to have the metabolism going. This is an example of a glucose pulse happening at MCS 300. And you see there's lactate production from the cells that produce lactate, and lactate consumption from the cells that consume lactate. So it seems like the metabolism is working as it ought to do. And this is another visualization just with all the different fields just for a shorter MCS. So we have most of the components together to actually start attacking our hypotheses. We weren't able to get the spatial arrangement, I guess, dynamics that we're aiming for, but I think we're really close to getting that. And as far as kind of like our stretch goals for this, I mean, there's a lot of different directions we can go, but I think understanding our original 3 hypotheses is kind of like what we're all interested in, and we're going to probably keep on working on this project and towards those goals after this hackathon. So I guess thank you all for taking the time to listen and hear about our project. It was a lot of fun doing this.
It was very ambitious. I'm really impressed how far you got in 3 days with that.
Thank you. Anyone ask any questions? Yeah, it's like we're hoping to get one or multiple papers out of this. I think naturally it's like if we go with the demineralization and the copper hypotheses, I mean, that's 2 papers there if the core model itself isn't a paper. So I think there's a lot to work on. And hopefully we can get the project in a more complete state. Yeah, thank you, Pedro. Yeah, if there's any more questions, I can answer them in the chat. Thank you all for the time.
Thank you. I think it's cool that you had 3 different hypotheses that can kind of be explored through the same general model structure. All right. In the interest of time, we'll go ahead and move on to Aritra, or jump ahead a little bit because it's stupid o'clock for Aritra where he is right now. So take it away, Aritra.
Screen is visible. So over the past few days, what I have been really trying to address is look into collective cell migration and how things like initial geometric confinement or cell substrate interaction affects it. So why collective cell migration is important? By this time, we all know it's a critical feature of morphogenesis and homeostasis. And typically, I wanted to replicate the results from this paper, which is from the lab where I did my PhD, where it was shown that cells are very sensitive to the curvature of geometric confinement they are in. So typically, there are 3 cases where the circle there is uniform curvature, and then you have a square and a cruciform where we have regions of sharp curvature changes. The experimental results show that cells do not deal with sharp curvature changes very well. So in regions where you have these changes, you see that there is less motility compared to higher motility where it's relatively flat or there is no change. And this is dictated by multiple things, which depends on cell-to-cell connection by this cellular contractility, how cells are adhered together by cadherin junction, so this leader-follower mechanism. So this is something I tried to understand using this, and these are the experimental data which hopefully the model can reproduce at some time, that how the envelope of the monolayer changes with time, how the displacement of the leader cells changes, how skewed they are depending on their positions, and the velocity fields. So this was my initial modeling goal. Some of this slide I presented in the hackathon initial talk. I mostly concentrated on doing the first one, where I tried to see how cells sense this geometric confinement, and can we simulate this with biological model, and then can we try to validate it. And then later, maybe we can try to extend it where we can incorporate cell substrate interaction. So this is very brief details about the model. I start with 3 cell types. There's the medium, then there are the cells, and there's an initial passive layer, which represents a geometric confinement. After some number of Monte Carlo steps, that layer goes away, and then we define the leaders depending on the cells that are in contact with the medium copper cells. The way the motility is done, I took a lot of influence from Corinne Bradstock, where she used external potential. So I have tried to use that, but change that thing a little. I have also used FTP-based links to ensure some leader-follower mechanism by using this, where I have a little bit higher lambda for leader cell than the cell to show that the leader to follower connections are faster. This is something people have also seen experiment. So this part is basically taken from Corinne Bradstock, but I also had a chat discussed with Pedro, where he suggested that the migration behaviors are regulated by this persistence effect. So I tried to incorporate that the direction of this force can be affected by this persistence parameter, where P depends on the weightage factor I assign to the old and the new. So if P equal to 0.8, then this is basically the persistence parameter. If I put 0.5, then this will be 0.5 of the old initial direction and the other direction of the velocity. So I tried to keep this around 0.7 or 0.75 for the simulations I showed, just to ensure that the cell migration had some persistence. And the curvature dependence was kept by a phenomenological term where I introduced this function f kappa, where kappa is a normalized curvature, and if this is large, then the motility forces decay. So this is something which is seen in the experimental results. It's like a phenomenological law that can hopefully capture. So this is what I did. I'm just going to play the video. So initially, this blue layer shows the confinement and the cells are inside. After some number of Monte Carlo steps, the lid is taken off and you see the leader cells forming, which are in the red. So what we see here is I tried with different aspect ratios. So for the ellipse around these, the curvatures are much higher. So here we should expect the cells to move more in the lateral direction than in the top. If I just play it again, you see that kind of a behavior is captured, where these cells are migrating more sideways. And as compared to the circle, it is pretty much isotropic because there is aspect ratio is 1 and there is no curvature, which is a constant. So then what I try to do is I try to plot some results. So I took 10 leader cells that are along this periphery at different angles, and I try to plot their trajectories. So what I see here is for the circle, it's mostly all of the cells move sort of the same distance. This trajectory, these are all in pixel units. But as you can see for the ellipse, we see that the ones which are in the flatter region, less curvature, they tend to move much more than the ones at the top, short curvature. This is much more pronounced as aspect ratio. So this is something we could capture. Of course, we have to validate, fine tune it further to validate with the exact experimental results and try the other geometries. These are the corresponding leader radial distance. As you can see for the circle, it's pretty much same, but this band becomes much larger and larger as you change the aspect ratios, because these cells are at different positions on the periphery. So where they expect in the regions of higher curvature, they are less, they have moved less. The ones that are more on the equatorial region, they have moved higher. Also try to track for the leader speeds. It's a bit messy, the scatter plot, but again, one can see the y-axis and you can see that the range is much more larger. The width, the spread of the velocities are much larger as one increases the aspect ratios. So it kind of captures what we are trying to say. Of course, we need to fine tune it a bit more. Future goals are first is to validate this model, calibrate it to in the experimental data. But this is another one part which I wanted to discuss. And Maisha, the other person who was trying to work with me, put some thoughts into this. So if we can also incorporate cell-substrate interaction. So there are some experimental studies when you do study cell migration on soft substrates like hydrogels. These are not just elastic, these are viscoelastic. So can we try to incorporate like the FTP link like a Maxwell model? This is something I had discussed with the Professor Glazier, and he suggested that maybe you can numerically compute this parameter where you can incorporate stress relaxation. This is a very nice idea. I hope to explore it in the long term. Another thing, this is probably very ambitious. I don't know if it's feasible, but in cell mechanics, when we try to measure the cellular contractility, there are these models by David Ode, these motor clutch models, where it determines how the cell binds to the substrate. There are rate constants which determine this binding and dissociation which controls it. That's a very nice mechanistic model that can predict cell migration. If at some point that can be incorporated, I think that would be nice. So I think with that, I'll wrap up. These are the different resources, the papers and other things.
Great. Well, that's a lot. You've made a lot of progress, and I think you have a clear pathway forward. It's quite exciting.
Just see.
I do have a quick question if we have time.
Go ahead.
Aritra,
Yes.
do you know what is the biologically what is causing the curvature dependence?
Yeah, that's an interesting question. So the paper, the data that I refer to the paper, what they try to do is they try to attribute it to the actomyosin contractility as well as the cell cadherin strengths. So what the experimental data shows is if the experiments are done, okay, I think I have it here. Oops. So basically, if you treat them with some drugs, which like blebistatin, which is a myosin inhibitor, then you do not see the migration at all. So that is one important factor which says contractility is important. The other factor is, there are some cells like MDCK where the cell-to-cell junction strengths are pretty strong. So that kind of governs this mechanism. If curvature is sensing some more precise biochemical signaling, I don't know, I need to check. But there are people who have done a lot of single cell studies. Collective cell, there are a little less.
Great, thank you.
Thank you. Okay, I'll just stop sharing for now. And thank you again. I received a lot of nice guidance. I may reach out to some of you later again.
Yeah, that's fine. If people wouldn't mind emailing me their slides, sometime today we can put them up in the hackathon folder. So people can take a look at this in more detail later. All right, next is, let's see, subepithelial fibrosis. Marcel, are you here? Yes.
Yes, I'm here. I will share my slides and apologize in advance because they're not very fancy. So just a very quick overview of what I was hoping to do. Everyone can hear me, right?
Yep.
Was just to kind of extend an existing ODE model of innate immune dynamics in response to biomass inhalation and basically create a tissue layer that the ODE model could talk to in a very simple way, at least initially, and includes some cells like fibroblasts and myofibroblasts in a subepithelial layer, have ECM accumulate, and then cause the airway to narrow. There's more to it than that, but that's like the quick overview there. And my schematic, I knew it was wrong, but it was at least a place to start, just to get the general idea. I knew I wanted to have some sort of signal from the ODE that drives cell behavior, cell dynamics in the tissue layer. And as we were starting this project, I had kind of laid everything out in terms of components in the model. I knew I wanted epithelial cells to be healthy or damaged. I have that in the ODE model, so I needed to figure out a way to have the two talk to each other. I wanted to have damage seated in the epithelium and the tissue layer based on a signal from the ODE layer, for example. And fields and so on. Let me actually just show, I think it helps to actually see the structure here. So this was one of the things I was initially, I'm not a biologist, so it really helped me working in this medium to figure out where everything should be. So the structure here is what I initially hoped to produce is represented here. We have an outer boundary that just keeps everything in this
I don't think you're
subepithelial.
sharing the screen you think you are.
Did I not share my desktop?
We were seeing your
I think I
browser.
compulsively select the browser just from teaching. Okay. All right. Hopefully, can you see it now?
Yes.
Okay. So this is what it looks like. Just to get a sense of what's included. There's an epithelial layer, there's fibroblasts and myofibroblasts in the subepithelium, there's space below the epithelium, that is the airway. That's what we've got so far. And properties and rates that I hope to. So this is basically all the slides that I had before starting. all of the things that I wanted to see in the model. And now I can kind of go over what we accomplished. So Christian tried out the ChatGPT CompuCell3D tool to like just get an initial model. And so that was an interesting experiment. So it put all of the cells in there. It kind of created a structure. It was all done very differently than I would have done. So it made it kind of hard to interpret, but it did work a lot faster. So there was some time spent in trying to understand what it had done, but it probably would have taken me much longer to use, uniform initializer and XML and so on and so forth. So that was an interesting experiment. And what was accomplished was, some challenges that we had was keeping the epithelial sheet intact. I know initially the model had everything just bursting through the epithelium. So I experimented some different ways to keep that from fragmenting. That worked. These are all the behaviors that I was able to get in there. Most importantly, I was able to connect the published ODE model, which is somewhat complicated, with CC3D successfully. That was like my main goal here, was to get that working. And the next step is to just work on the biological realism and work on revisiting how I want to have the airway shrink, because there's several different ways to do that. And this is a slightly off topic, but I thought people would be interested because AI tools are something we've talked about a bit. So not only did we experiment with getting ChatGPT to produce the model initially, that was what Christian did. For me, something I did over the last, or basically over the summer, was I've been working on doing things like learning Unix shell again, learning Git, training, learning Claude code, training my own graduate assistant to be my record keeper, and being very careful about version control, model assumptions, keeping a lab notebook. So something very interesting, and this happened because I actually signed up for the hackathon last year after collecting 6 semesters of my own students' work and my notes, thinking that I'd be able to put it all together. Sorry, I'm making dinner at the same time. But I ended up having surgery that weekend, and then I had several surgeries over the course of the year, and all of this I've kind of forgotten. So I knew when I started this summer, I was going to need to have like a kind of an AI buddy help me sort through all of my documentation. And so this is just an example of how that helped me out here. When Lorenzo came to help me figure out how to get a cell behavior that I wanted into the model, it broke the model. And so I went back to get Claude to help me diagnose. And the interesting part was how it could remind me, hey, this wasn't in your lab notebook. Originally your assumptions were to do this other thing. Should you be doing this in the first place? And so I think this has helped me stop drifting off. I keep pausing and going in there and saying, hey, look at my lab notebooks, look at my original model assumptions. Have I diverged from my intentions? And it can point that out to me. So I think that's like, I don't feel comfortable having it do everything for me, but I think it is really helpful to be my second brain. So I just thought I'd share that experience. Future work. I definitely need to redraw my schematic, because something I've found makes me really uncomfortable is working on the model with the wrong schematic and not really having a good sense of how what ChatGPT or I have put in the code relates to my original intention. So that really needs to be done. But practically speaking, model outputs, I have several different ways. Like I've gone through published work and I've seen there's a couple of different ways that people actually quantify airway shrinkage. So I need to figure out which one is easiest to get from CompuCell3D. So that's something I'd like to do next. Run some simple experiments, and then ideally publish, and also figure out what sort of in vitro wet lab experiments my students can do that could connect to this, because I would like them to be able to, learn sufficient CompuCell3D to do parameter scans or just play around with the model. But I'd also like them to be able to collect some data as well. You might be missing a closure slide. Yep, that's where it ends. Thank you so much, though, for your help. I can't believe how much was accomplished in this time period. Christian, did you want to add anything?
I think Christian has stepped away from his computer and forgot he was logged in.
Okay.
Well, thank you, Marcella. That was really impressive. Really appreciate all the work and coming back a second time to do it. And that AI assistant is exciting as well. So I think you could teach us a lot too, probably.
Yeah, actually, it's the third time, right? Because I actually I was in the 2023 workshop and built an M1, M2 polarization. And, that's why I did the AI tools, because I'm getting tired of relearning CompuCell3D. I want this to be the last time. But yeah, I'm really looking forward to forming connections and getting continued help, because I would love to publish in that special issue in May.
That would be fabulous. Hope we could do that.
Great. Thank you, Marcella. We did tumor growth reproduction. So we've got, let's see.
Why don't we do bone cell dynamics next?
All right. Gabrielle, are you there?
Yes, hello. Let me share the screen. Okay, let's go. Our main objective was to build a bone-on-chip model that combines scaffold geometry, perfusion, oxygen transport, and cell mechanics. So the title was Oxygen Heterogeneity and Bone Cell Dynamics in a Perfused Bone-on-Chip Scaffold. We want to understand how some perfusion rate affects osteoblast and osteoclast transport, retention, and fate inside the scaffold. In practical terms, we are asking whether cells remain near the inlet, penetrate the porous scaffold, or are eventually washed out through the outlet. So the central question is, can the scaffold retain cells while still allowing sufficient oxygen delivery through the pore network? For this first prototype, the scaffold is represented as a two-dimensional static gyroid-like section, and the scaffold itself does not remodel during the simulation. Can you see the second slide?
Yes.
Okay. So the flow is in the low Reynolds number. We use one-way coupling, meaning that cells respond to the flow field, but they don't modify the fluid dynamics. And oxygen transport occurs only in the pore space, while the scaffold is treated as oxygen impermeable. We initially seed 200 osteoblasts and 200 osteoclasts and simulate 24 hours using 10,000 Monte Carlo steps. Cells that reach the outlet are counted and removed from the domain. The cell behavior described using the cellular pods model. This effective energy contains several contributions. The first term describes contact energy between neighboring cell types. Then we have volume and surface constraints, which control cell size and shape. We also include a connectivity term to prevent cell fragmentation, and an external potential term associated with flow-induced motion. and an adhesion term for molecular cell scaffold interactions. So cell movement results from the competition between these energy terms during the Monte Carlo updates. Osteoblasts and osteoclasts also have different target volumes and surfaces, reflecting different coarse-grained cell sizes. For the transport physics, we use a pre-computed low Reynolds number using Navier-Stokes equations for the flow field. Oxygen is then described by an advection diffusion reaction equation. The advection transports oxygen with a local velocity field. Diffusion redistributes oxygen spatially, and the reaction term represents cellular consumption. The cellular oxygen uptake follows Michaelis-Menten kinetics, so consumption depends non-linearly on the local oxygen concentration. In this model, these were the diffusivity coefficient and the Michaelis-Menten constant. The model was implemented following these statables and functions. For cellular interactions, we use two complementary mechanisms. First, the standard contact plugin defines baseline interfacial energies between cells, medium, and scaffold. The adhesion flex adds an explicitly receptor ligand adhesion layer. Finally, when a cell is touching the scaffold, its flow-induced drag is reduced. This represents mechanical retention due to scaffold contact and adhesion. Here we can see the actual simulation domain. Just a moment. On the left is the initial cell seeding and inlet region. The blue cells are osteoblasts and the green cells are osteoblasts. The light seen structures are the solid scaffold, while the black interconnected region represents the pore space containing the culture medium. Oxygen and perfusion enters from the left and move toward the right through the pores network. Therefore, the geometry creates multiple possible pathways and local regions where cells may either be transported or retained by the scaffold. Now I'll show the simulation dynamically. We have the cell field, the flow speed, the oxygen depletion, and then oxygen concentration. Let me know. Okay. The important point is to compare where cells move with the local flow pathways. We can also observe where cells remain retained near scaffold structures and where they progress downstream. At the same time, oxygen depletion becomes stronger in regions with cellular consumption, producing spatial heterogeneity in the oxygen field. So flow organizes transport and oxygen exposure, while scaffold contact controls retention. And we can also quantify how many cells completely leave the device. I think in this simulation were only 2 cells that complete. And as future work, the next step is not simplified to choose parameters manually, but to perform a systematic sensitivity analysis. We want to answer practical questions such as what fraction of cells should remain adherent to the scaffold, what mean oxygen concentration should be maintained, what fraction of dead cells is still acceptable, and what maximum fraction of cells should fully cross the devices. We will first use the Morris method for global screening to identify which parameters actually influence these outputs. Then, for the most important parameters, we want to use Sobel analysis to quantify their individual effects and their interactions. This will guide target calibration in future scaffold or operating condition optimization. So this model gives us a framework to connect scaffold architecture, the perfusion, the oxygen availability, and cellular retention in a single bone-on-chip simulation. And to finish, I want to tell you that I'd like to express my gratitude for these days at the workshop. It has been a very enriching experience and a great opportunity to learn the fundamentals of CompuCell3D. And I'd also like to thank you all the instructors for their time, their support, and everything they shared with us. And thank you very much.
That's really impressive. You got a lot done. Made a lot of progress, and seeing that explicit flow model is quite interesting. I hope we'll have opportunities to work with you going forward as you develop that.
Thank you. I hope to.
All right, thank you, Gabrielle. And then finally, we have our hybrid CPM group.
All right, can you see my screen?
Yep.
Looks great. Thank you for being willing to be the last. Definitely
Yeah.
not the least.
So, yeah, we were, I'm Levi. My teammate was Leonardo. We set off to be able to develop this hybrid cellular POTS and molecular dynamics model of collagen fiber networks within CompuCell. Oh, hold on. Okay, so yeah, the aim and context, collagen fiber networks and cells have this dynamic interaction between each other. And so being able to model that mathematically within CompuCell would be advantageous to being able to kind of see how cells and the collagen fibers interact in various environments. And so we set off to replicate the Kaiser et al. Paper published in 2025. And so in order to be able to do that, we would need to develop this plugin, which we had started trying to do, but that ended up being a little more extensive than we had hoped for this hackathon. And so we shifted into seeing if we could get just molecular dynamics working within the steppable using the Python package whomd blue. And trying to get that integrated in with the CPM model itself. And so going over our implementation, first we initialized this extracellular matrix through these beads. And so you can see back here in the panel on the right, you have these different colored beads. And so there's, or like particles that represent the collagen fibers. And so what we started out with was just this grid of beads that we were hoping to be able to connect horizontally and then vertically to represent just like a grid of collagen fiber. And so at each MCS, we start with our cell and obtain the cell pixels that are overlaid with a bead. And so then if a bead lies within the cell pixel, you label it as this adhesion bead. And then that is then connected via a harmonic bond in HUMD to the cell center of mass, which is calculated through CompuCell. And then at each MCS step, as the cell moves around, the cell kind of will start to overlap with additional beads that were not initialized in the beginning, and then we update the bonds. And so here, so with our simulations, we did start with kind of the example that Michael had when he was showing us how to do plugins. And so instead of the Y shape, we have this initialized grid in red. And so the cell is going to want to kind of adhere to this, to the red structure grid. So in this simulation here, you can see our cell is initialized, and then all the particles that exist within that cell go to the center of mass. And then as the cell kind of starts to move over additional beads, those beads are then dynamically pulled in to the centers of mass. And so you can see kind of there, here. Not sure if there's any more in this video. So yeah. And then, yeah, so Leonardo is going to kind of summarize what we did and some next steps.
Yes, thank you, Levi. So concretely, what we did is use the plugin that is native to CompuCell that tracks the pixels that belong to a cell, and with that there was some sort of adjustment of units of measurement, because we had to convert from the CompuCell3D pixel grid into the one that is used by the package UMD Blue. And so like making those match up, we were able to run this loop that checks if the beads are overlapping with the cell. And if that was the case, then we also implemented these effectively they are spring potentials that link the beads in the ECM with the center of mass of the cell. And this is due to a tension force due to the cytoskeleton of the cell. And that causes the sort of the conversion lines that you see in the simulation. And we also set up the other elastic potentials that are described in the 2025 article, namely the linear, geometrically linear potentials between the different beads, and also the angular potential between triplets of beads that sort of like tends to one making the fibers to want to straighten out. And yeah, so the future steps are implementing those potentials as well, and initializing the ECM in different configurations to test the hypothesis on the strength and stiffness of the ECM and how that changes the behavior of the whole model. Yes, that's really it.
Well, that's a lot. That was a very ambitious project. Seeing you be able to couple to the particles is exciting. I hope you'll feel inspired to keep going on this. That would be fabulous.
Yeah, it'd be cool to be able to fully develop that plugin.
Yeah, we'd definitely be happy to keep working on that with you. I know Michael's pretty interested in continuing on with this potentially. TJ Sago might also be interested in some of this. He's one of our previous collaborators and developers of CompuCell who actually built or helped build a package called Tissue Forge, which is a more it's center-based molecular dynamics approach to similar kinds of simulations. So there could be some potential to pair it to Tissue Forge or potentially to have TJ take a look at some of the work you've done this weekend. I can make that connection for you guys in the next few days if you'd like.
Yeah, that'd be great.
Yeah. Okay. All right. Well, let me share my screen one last time for a thank you to everybody for sticking out both the workshop and the hackathon. We know that it's a lot of time and a lot of material. So we really do appreciate everybody spending their time in the summer with us. In this way, I'd like to thank all of our instructors who helped out over the course of the weekend with the hackathon, as well as everybody here who participated. And as we've mentioned before, if anybody would like to continue working with us on these projects over the next weeks or months, please feel free to reach out and we'd be happy to set up some meeting times with you. So yeah, just let us know if you need anything, and we look forward to hopefully seeing some of this work either published or put up on GitHub or whatever over the next year or so, or however long it takes.
I really want to thank everybody again for all the great projects and the work this weekend. I know it's a lot of time, and if you're in a bad time zone in particular, so I really do appreciate those of you who stuck it out to the end. It's been fun working with all of you. And I want to thank Hayden for doing an amazing job organizing this meeting this year, and to Gilberto, Pedro, Lorenzo, and Michael for helping to teach and for TAing. And with that, we are done for the 2026 hackathon.
Yep. Everybody is finally free to go get some rest.